discuss question answers of Unit 3, June 2011. We will try to cover as many as papers possible for this unit. So let's begin. The 5 prime cap of RNA is required for. So the 5 prime cap of RNA, it is required for the stability and transport of RNA. If stability is not there, this will be degraded in the cytoplasm. So to move this RNA, mRNA into the cytoplasm uh, and to make it stable, this 5 prime cap is required. The answer will be B. So coming to the next question. A culture of an E. coli strain that is lysogenic for fudge lambda, it is grown at 32 degrees centigrade, and induction of profudge, which is the integrated fudge, from the host chromosome will occur when the culture is exposed to. So it is saying that a culture of E. coli strain for fudge lambda, it is grown at 32 degrees centigrade, and induction of the profudge from the host chromosome will occur when the culture is exposed to. So induction will occur when the culture is exposed to UV radiation. So in case where there is UV radiation and where there is like action of certain chemicals, in that case what happens? So when there is UV radiation and certain chemicals, in that case there is the induction which will occur in presence of certain chemical. Just a moment. So what will happen? This induction will help in release of the bacteria, release of the virus, release of the virus, and the virus will be released as they will undergo lytic cycle after induction. So this is due to ultraviolet radiation. Next is the fidelity of replicative base selection can be reduced by a factor of 10 to the power 2 when the repair of DNA synthesis involves. So fidelity, what it can be reduced by a factor of 10 to the power 2 when the repair of DNA synthesis involves. So when the repair of DNA synthesis involves uh, involves which of the enzyme will the fidelity will be reduced? So fidelity will be reduced when like TLS DNA polymerase will be used. Okay, when TLS DNA polymerase will be used, then the fidelity of the replicative base selection can be reduced by the factor of 10 to the power 2 when the repair will involve TLS DNA polymerase. Now, why is it so? This TLS DNA polymerase, it is translation DNA polymerase. It is translation DNA polymerase, and this translation DNA polymerase will reduce the accuracy. Fidelity means the accuracy to replicate the template, and this TLS polymerase, they are the family of Y family DNA polymerase, and they are kind of unique class of DNA polymerase which have specialized structure which allows to repair damaged DNA substrate. Okay, so it generally leads to mutagenic DNA synthesis. Okay, low fidelity polymerase. So question is, what is the minimum number of NTPs required for the formation of one peptide bond during protein synthesis? So for the formation of one peptide bond, number of NTPs required is 4. Number of NTPs required is 4 for one peptide bond formation. So the next question, coming to part C. Lac repressor inhibits expression of genes in lac operon, whereas purine biosynthesis is repressed by I repressor. The two proteins have 31% identical sequences and have similar three-dimensional structures. T gene regulatory properties will be the gene regulatory properties of these proteins differ in relation to. So lac repressor is inhibiting the genes in the expression of lac operon, whereas purine biosynthesis is repressed by say some kind of gene you're printing mistake, and the two proteins have 31% identical sequences and have similar three-dimensional structures. Okay, which two proteins? That one is purine biosynthesis repressor and another is lac repressor. Okay, and the gene regulatory properties of these proteins differ in relation to. So binding of small molecules to the repressor, if you have read about lac operon, you have understood that when allolactose binds to lac repressor, in that case repressor cannot bind to the operator, as a result of which operon will be on. So small molecules when bind to the repressor, this is correct. Next is presence of recognition sites on the genome. So there must be some recognition sites in the genome. Then oligomeric nature of the repressor. So because there is a similarity between these two, so what characters of the one is similar to the other. Okay, so also repressor is kind of repressor is oligomeric in nature. When you have understood about the repressor, it's like oligomers are there. The lac repressor is a oligomer. So S is DNA binding property. So DNA binding property might not be true. So according to me, this small molecules is definitely correct, as allolactose is also the one which binds to the repressor. Recognition sites on the genome and oligomeric nature of the repressor. So P and R is definitely correct, but here Q will also be correct. So answer will be B. Next question is two E. coli cultures A and B are taken. Okay, two E. coli cultures A and B are taken. Culture A was earlier grown in the presence of optimum concentration of gratuitous inducer IPTG, and both the cultures are now used to inoculate fresh medium containing suboptimal concentration of gratuitous inducer. So culture A was earlier grown in presence of optimum concentration of gratuitous inducer IPTG. Now both the cultures A and B, now in this case both the cultures A and B, they are now used to inoculate fresh medium containing suboptimal concentration of gratuitous inducer. So this is a fresh medium and this already have suboptimal concentration of gratuitous inducer. It was observed that culture B was unable to utilize lactose. Okay, B where no initial IPTG was there, it is not able to utilize the lactose, whereas culture A did so efficiently. The reason behind this is, so the reason behind this is, uh, when IPTG is there already there, in that case what will happen, there will be the induction of lac permease. Okay, lactose, like minimum level of lac operon will be on, as a result of which when lac operon will be on, so gratuitous IPTG is when it is there, operon is on, and when operon is on, ZYA will be synthesized. There will be suboptimal concentration of Y gene will be active. This Y gene, which is lac permease, it was induced to a high level, okay, during pre-treatment. Its level is high during pre-treatment, which allows the uptake of lactose from the fresh medium. Okay, so when GI, gratuitous inducer IPTG, is added, it allows the the expression of Y genes. So more permease, more uptake of lactose will be there. So this is written in number C. So it has been observed that 5 to next question, it has been observed that in 5 to 10% of the eukaryotic mRNA with multiple AUGs, the first AUGs is not the initiation site. Okay, for some AUG is not the initiation site. In some cases, in such cases, the ribosome skips over one or more AUG before encountering the favorable one and initiating translation. So this is postulated to be due to the presence of following consensus sequence. So which consensus sequence? Here it is saying that ribosome skips over some AUG before encountering the favorable one and initiating translation. So which of the following consensus sequence is there? Okay, uh, like, so for initiation, the ribosome will find out first AUG, which is in the consensus sequences, which is if it's saying about the eukaryotic mRNA, they look for Kozak sequences. So the AUG which is not present near the Kozak sequence is skipped, but the ones present in the Kozak sequence, which is CCAUGG. CCAUGG, this is a Kozak sequence. So the one AUG present near the Kozak sequence is will be the first initiation codon. So this is the consensus sequence, Kozak sequence. Okay, which is the initiation site in most eukaryotic mRNA transcript. Clear? So answer will be P and Q. So next question is, presence of circular mRNA for a specific protein in a eukaryotic cell reflects a rapid rate of synthesis of that protein. Okay, circular mRNA reflects a rapid rate of synthesis of that protein. Following mechanisms are suggested. Which of them is correct? So EIF4G and PABP, they promote this process through 5 prime to 3 prime interaction of the mRNA. So first of all, let's try to understand the function of EIF4G. PABP. So this EIF4G, EIF4A, EIF4B, okay, G, A, E, B. So all, what is the role of all this? Okay, EIF is eukaryotic initiation factor. So translation initiation in eukaryote, it requires the involvement of multiple factors, which is eukaryotic initiation factors, and all these factors they facilitate the binding of the 40S ribosomal subunit. What they do, they facilitate binding of 40S subunit to mRNA. Okay, so that this can form 80S. So that they can form 80S. Okay, so this EIF4, okay, EIF, EIF4F, the EIF4F, this consists of 4E, 4A and 4G. Okay, and this EIF4F, it binds to 5 prime cap. 4F will bind to 5 prime cap of mRNA, and it helps in the recruitment of 40S subunit. Okay, and helps in recruitment of 40S subunit. Okay, EIFB is not mentioned here, but EIFB, what it does? EIFB, what it does? It promotes the ATP dependent RNA helicase activity. Okay, helicase activity of EIF4A and EIF4F is promoting the activity of A and F. Okay, and this activity is required to unwind the secondary structure present in the 5 prime region. Okay, otherwise this will impede, this will enter into the 40th subunit. Okay, another protein, poly A binding protein, which binds to poly A tail. This binds to poly A tail. Okay, and this poly A tail with binding protein plus EIF4G, 4B, 4A, they what they do, they promote circularization of the mRNA and promote the translation by 40S. Stimulates translation. Stimulates translation by recruiting 40S. By recruiting 40S. Okay, so these factors are essential for rapid rate of synthesis of the protein. So here also EIF4A also promote this process, and 4G like 4G, F, A, E, B all are important in playing this role. Okay, so for that C is correct option. Next question is SI and miRNA are used for gene silencing. The major steps are similar. There are distinct differences in the key players of the two processing pathway. Processing pathway are different, distinct in processing. Following statements are related to some characteristics of gene silencing. Which of the following combination is not correct? So both siRNA and miRNA, they are processed by cytoplasmic endonuclease dicer. So both of them, are they like processed by dicer? What do you think? No. The processing pathway is distinct as it is mentioned in the question itself. So this becomes wrong. Drosha is needed for processing miRNA and precursor siRNA. Okay, so for processing, drosha is required for miRNA and precursor siRNA. So this is also wrong because same drosha is required only for miRNA. Next is both Si and miRNA, they show association with Argonaute protein. So this is the one which plays role in cleavage. So this is required for both. Next is both the processing pathways involve risk complex. So this is also correct. Risk complex is involved in both cases. Argonaute protein is involved in both cases, but P and Q are wrong because dosha is not required for both and processing is different in both the cases. So sometime in some of the question when we will be doing with the part C questions where I will be explaining you miRNA and siRNA differences. So for now, C is the incorrect option. So the last question for June 2011 is genetic studies demonstrated that TBP mutant cell extracts are deficient in transcription of genes. So when TBP is mutant, transcription uh, they are deficient in transcription of genes from all three promoters. Okay, class 1, class 2, class 3. If TBP mutant cell extract do not have these promoters, okay, do not have these promoters, I said wrongly, that means they will not show transcription from any of these promoters when there is TBP mutant. Okay, so following statements are described characteristic features of TBP. which one is not correct. Please, whenever there is not mentioned, but we only look the answers for the correct ones. You have to read the question very carefully. So TBP is considered as a universal basal transcription factor. It's true. It's transcription binding protein. Okay. So this is the universal basal transcription factor considered. Okay, this is true. So wherever P is there, please cut option elimination, because not correct is asked. Next is TBP is not required for transcription of archaeal genes. So is this true? It is required. Why not? It is required for the transcription of archaeal genes. Archaeal genes also kind of have the same mechanism. So this is wrong. So wherever Q is there, you cut that out. This is wrong means this should be included in your answer. Okay, so Q should be there in your answer. So this also got eliminated, but only S remains. But let's read. TBP is involved in recognizing Tata box. This is correct. Okay, so this should not be there in your answer. Next is TBP operates at all promoters regardless of their Tata content. So it can Tata content is very important for the TBP to operate. So if Tata content is not there, TBP will not operate. So this is also wrong. So your answer will include two incorrect statement, which is Q and S. So that's it. Next we will come with December 2011 paper.